All right, hello everyone. Like Angela said, my name is Liwski. I led the team that developed the ReSeq TB platform. We actually started in earnest probably about a year ago now. So the first year we spent a lot of time figuring out which data sets we wanted, figuring out how to structure the data in the database, figuring out the mechanisms we needed to put in place for contributing data and things like that. So our first year has really been focused on getting data into the data platform. And one of the things I want to, that will be an ask of my session today, is I'd really like to get feedback from people, the users, on now how do we make the data useful to researchers, to diagnostic developers, and ultimately to clinicians. What I'm going to do is give you kind of a high-level overview of all the different facets of the data platform. So I'm going to talk about data acquisition, the incoming data repository, the investigational database, which is where we collate and aggregate and normalize all the data, how users access the data today, and then I'll talk a little bit about futures. All right, so first, data acquisition. All right, so this is kind of a high-level schematic of the way the data platform's structured. Jamie's already talked about the unified pipeline, so I'm not going to talk too much about that. But then we have a lot of different kinds of contributed data. Primarily today, the contributed data has been genomic and phenotypic data, which is what we want. We've got some leads on getting data sets with more clinical data, but there's not a whole lot of clinical data in there now, not a whole lot of outcome data. That's what we hope to get over the next year. So that data goes through an expert panel review. We have several people from around the world who are looking at data sets that we want to acquire and analyzing the quality of those data sets. Goes through a curation process once it gets into the Critical Path Institute. Goes into an incoming data repository. One of the unique requirements about the ReSeq TB platform is not only did we want to make the aggregated data available to end users, but there was a requirement that end users could actually access the individual data files as contributed. So that's what the incoming data storage server does. And then that investigational database, that's where we aggregate the data. It goes through a data mapping process where we normalize it to a standard. We use the CDISC standard. And then we make it available to end users. So first we'll talk about the contributed data and how it gets to the incoming data storage server. What I'm going to focus on is the actual technological processes we use to transfer the data into the incoming data storage server. There's a lot of work that goes on in finding the right data sets that we want to go after, signing data contribution agreements, going through legal agreements, reviewing the quality of the data, finding out gaps, things like that. What I'm going to talk about is actually how we transfer the data into the incoming data storage server. So we've got three methods today, and the first method is the one that we use primarily. Jamie talked about how everything we've gotten that they've sequenced today has been available in NCBI, SRA. So the first method we use to get data into the platform, at least the genotypic data, is we download it directly from a public repository. And that's a very convenient method. It's nice because somebody doesn't have to upload the data twice. But when we first designed the platform, we envisioned that people would transfer the data directly to us or upload it directly to the platform. So we have methods to support that as well. Here's just a screenshot of one of the SRA transfer screens. And then the secure FTP server is another means we've used, and we use this a lot to transfer the phenotypic data. Those tend to be smaller files. Our history at Critical Path Institute has been with clinical data, so we have a very stringent security policy on transferring the data, and the secure FTP server, which we call Crush FTP, supports that mechanism. I've got a screen interface for that. It's nice because it works on any web interface. If the transfer gets interrupted in the middle of transferring these very large files, it will start back up when you reconnect. And then the last method is the one that we hope to progress in the future. And this is where end user would actually upload the data directly to the ReSeq TB platform. It's actually working today. We've used it to transfer data from the FIND group, and that's one of the things we hope to enhance over this next couple months to make it more user-friendly for others who can contribute data. And basically, you've got a drag and drop interface. You log into the ReSeq TB platform. You drag files from your hard drive or from an FTP server, and it can transfer, I think, up to 100 files at once. Now, some of these are very large files. That could take overnight if you're transferring that many files, but nonetheless, it will run the transfer for you. You can walk away and it'll move everything for you. All right, next I want to talk about the incoming data repository. This is where we store all the data as contributed, the individual files before it gets mapped and aggregated. And this was, talk a little bit more about this. This was a challenge because we get thousands and thousands of files. So the way we've structured the incoming data storage server, we have everything separated by projects. So you'll have a set of project files which is really tied to a specific study or a specific contributor. You can actually filter off of projects within the aggregated database. So if you want to tie some analytical finding from the aggregated data platform to the source file data, you can look at the project and bring it and come back to the incoming data repository to find the source file for that data. Within a project, there's this file management interface, and this is one of the enhancements we're working on now, because one of the things we realized after we started loading all this data is when you've got 2,500 SNP files or 2,500 whole genome sequence files, and just listing them out in a big long file list makes it hard to navigate. You can filter the files, you can do searches to shorten that list of files, but we're working on an interface now where we based on category, you will have an expandable list if you just want to look at SNP files, if you just want to look at the whole genome sequence files, if you just want to look at the output of the pipeline, things like that. All right, next we'll talk about the investigational database, and there's a lot of activity that goes on between data coming from the incoming data repository to when it goes into the investigational database, and that's where the curation and mapping happens. I'm not going to talk too much about the mapping process, but talk about how it gets into the investigational database. And that's this section over here with the multicolored bands. And the multicolored bands, what they represent is we've got a hierarchical access process within the data platform. The idea behind this access process was we could have different tiers of access for different users depending on what they needed to do with the data. What we're actually going to do with it, where it becomes most useful, is if we ever need to embargo data. Somebody wants to upload data to the platform, but they don't want to make it generally available for a year or something like that. What we can do is load the data. We can make the summary data available in aggregate, but we can restrict access to the actual incoming data storage, and we can actually restrict access to the individual files when you do a browse of the data in the aggregated data platform, in the investigational data base. A little bit about the process we use to map the data. So again, our history, a lot of our history is with clinical data, and when you get clinical data from different contributors, no two data sets ever look the same. And so there's a big process we go through to review that data and map it into a standard, a CDIS standard. And as part of the development process for ReSeq TB, we were actually in the midst of going through an update to the TB standard, and it was very complementary, actually. I think we worked with our standards team to understand the gaps in data contributed versus how we needed to map it into the investigational database, and they used that to build that TB 2.0 standard. The nice thing, or actually one of the surprising things about the data in ReSeq TB, it's primarily genotypic and phenotypic data, and because we control the pipeline, the genotypic data is already pretty much standardized when we get it. We have very little mapping that we need to do. Matter of fact, we've worked with Jamie and Matthew to structure the data more conveniently so that it makes it easier to map it into the data platform, and they've been great and turned around changes very quickly. So it's very nice now. Mapping for genotypic data is very quick. We can do it very quickly. We have a program we developed to do that transformation of the data, and usually when you get data from different contributors, you have to keep changing that program to tweak it a little bit for each contributor. With this platform, we have the same program. We run it over and over again. Phenotypic data is a little bit different, but it's also much smaller. The SDTM standard, the CDIS standard for phenotypic data already existed, so that's pretty much a well-known entity, and the mapping is fairly simple for that as well. As we get into more MIC data and critical concentration data, we'll have to do a little bit more transformation, but we understand how to do that, and that shouldn't be a problem either. All right, now let's talk a little bit about data access, how end users actually use the data platform. Today we have about 30 approved users. Most of them are CPTR members, made up of researchers, DST developers, and then analytic bioinformaticians. Everyone accesses the data the same today. One of the things we have talked about is creating separate interfaces depending on the research needs. If a diagnostic developer needs a very simple list of mutations associated with resistance, we can give them an interface, and then if the researcher needs access to more detailed data, we can give them an interface for that. Right now, everybody accesses the same interface, and there's 3 different methods for getting to the data, and I'll show those here. First one is a data browser, so you can actually view the data online based on what we call domains, but basically that means you can look at all the phenotypic data in aggregate, and you can look at all the sequence data in aggregate, or the genomic data in aggregate. It's nice if you know what you want to look for. It's nice if you want to look for certain peculiarities within the data. What it's not convenient for is if you want to associate the genotypic data to the phenotypic data, because in this browser view, you're looking at it separately. So what we also allow is a data export. You can download it in SAS format, or you can download it as a CSV file. And today, that's probably the most prominent method that researchers would use. They would download the entirety of the data and then use their own analytical tools to perform whatever analysis that they want to run. And then the final way is we have these data analysis reports. So we have 2 drug-resistant mutation reports. One's a simple version and one's a more complex version. I've got a sample of one of those. And this, again, is another place where I could really use some feedback. We've worked with the expert panel to define the format and the content of these reports. But if anybody notices something else that they like, we've already gotten some feedback from several people that we'd like to add to the reports now. That's the kind of feedback I'm looking for, is what else do you want to see in these reports? We can create as many reports as we want, as we need to, or we can enhance the existing reports. This is a snapshot of the simple report that we have now that just shows by drug and by mutation what the association and confidence levels are with specific resistance patterns. All right, talk a little bit about the future. So a lot of this is kind of motherhood stuff. We're going to go through ongoing maintenance and expansion. We're constantly monitoring system performance. Some of the current statistics. So we've been live since October 30th of 2015. We've had about 1,000 accesses, or different user accesses so far of the data platform. And we're seeing anywhere from 80 to 100 users per week. And that represents at least 10 countries. The problem with the analytics is if the browser doesn't say which country it's coming from, it just shows up as unknown. So about 1/3 of our accesses are from an unknown country. That could be anywhere. But the prominent accesses are from European countries and the US. And actually South Africa is one of the higher ones as well. And I think somebody mentioned before, we have about 3,500 isolates in the database right now. So we'll continue to monitor the access. Right now, one of the things we were worried about was whether the performance of the data platform would be sufficient to support the users. Right now, everything's fine. We haven't had any bottlenecks. If we get to the point where more and more users are downloading these large sequence files, we may have to expand the interface. It's on an Amazon cloud right now, so that's very simple to do. Probably our biggest focus right now is still collecting data, mapping the data, and uploading it into the data platform. We do have a plan, and part of our grant was to perform an assessment of the data platform. What we've got built into the platform, there's a feedback button on every single page. And again, for anybody using the platform, I encourage you to just use it. It's a very simple interface where you say give feedback, lets you say whether you found a problem or have an enhancement suggestion, and then it gives you a big text box to enter your request. That comes directly back to us, and we put it into a tracking system for a future enhancement. And then we'll conduct, once we have a pretty good population that are using the data platform, we'll actually go out and have individual calls with users to query their feedback, and we're meeting with the expert panel this week to get some feedback from them as well. But I want to focus on this third bullet here that says enhanced platform capabilities and user interface. I've just got a simple bullet here that says based on the assessment from users. But this is, again, where I'd like feedback and where we really want to focus our efforts on this year and next year is building in those analytical tools that end users need to perform the work they need to do. Some of the things we've already worked on, we've got a multi-user landing page now to make it a little bit more globally accessible and understandable. We're working on an interactive user report interface. Basically, that simple report that I showed you, we're going to build that into the platform so you can do filters and queries off of it. Some of the other things we've talked about, we're very interested in understanding what kind of analytics tools end users want to see, and developing analytics tools from scratch could be cost prohibitive. If we can integrate something that exists, that would be perfect. If we could export the data for use in another analytics platform, that would work out great too. But if there's a simple interface we can add or a modification of an existing analytics tool that we can integrate into the platform, that's what we'd love to hear. That's the kind of feedback we'd like to get. The other big thing we're working on, which I kind of touched on, was the organization of the data on the incoming data repository. Right now, just seeing a list of 2,000 files probably isn't very helpful, even if you can filter off it. So we're going to build in some tools to help navigate that a little bit easier. The other thing we've talked about, there's two other things we've talked about. They're not in plan, but again, I'd like to hear feedback on the prioritization and urgency for doing things like this. One is building the pipeline into the platform. It actually works out quite nice the way we're doing it today, but we certainly see an advantage to being able, especially when we have most users uploading data directly to the platform, to be able to do a simple submit and have it run through the pipeline and actually take the output directly into the incoming data repository. That's probably not too terribly difficult to do, but until we have a very stable, until we've gone through a couple iterations of the pipeline, I'd probably want to hold off on that. The other thing we've talked about, because the expectation is, or at least the feedback I'm getting, is that a lot of users may want to download these very large sequence files. We're talking about working with NCBI to create a bio project where rather than have them download everything from the ReSeq TB platform, we could link to the existing bio project, give them the accession numbers, and they can download it directly from there. And our goal for towards the end of this year is to make the tool available to, right now it's kind of internally available to CPTR members as we get feedback. October, I think, towards the end of this year, we plan to make this available to any research with a qualified research project, and I'd like to have as many of those analytics tools, interactive reports there as possible. Here is a snapshot, and I think, Max, if you can jump to our web page.
We're on it.
Oh, can you scroll up? So here's the actual live page today. I thought it'd be more interesting to show. And so you can see the scrolling welcome screen. There's actually a drop-down menu at the top to select a language, and you'll get the introductory paragraph in up to 6 different languages. Do we want to do that's I think everything I wanted to cover. Angela, do we want to do questions now or wait till the end of the next? All right. Well, thank you all very much.
Thanks, Rick.